Now that there are 3 cases of the Omicron variant in Austin, we're taking a closer look at how variants are discovered. The KVUE Defenders dove deep into how that works. Investigative reporter Erica Proffer explains.
So we have tubes.
When we take a PCR COVID test, the test ends up in a fridge like this.
Each of them has a barcode.
Virus killed, but sample preserved.
Remove the cap from these tubes.
This is at the University of Texas at Austin.
And transfer them
A small sample from the test goes here,
into the robot.
so it can be sent here, where chemicals help scientists look for the SARS-CoV-2 virus, the virus which causes COVID-19.
All of the stuff that's in saliva that is not RNA, we want to wash away.
After some mixing here and here, the samples go here.
Transfer the plate into this instrument to get the results.
This machine looks for the SARS-CoV-2 virus.
This is a positive.
What happens next can change the way we see the virus altogether.
All right, so this is a flow cell.
This is the first step in hunting variants.
It tells you what you put in there.
It's called genomic sequencing.
All life on Earth has to have a program that encodes in its genetic information, the genome. For many organisms, that genetic information is DNA.
Dr. Ilya Finkelstein is an associate professor of molecular biosciences at UT Austin.
Some organisms can transmit their genetic information via RNA.
SARS-CoV-2 is made up of a strand of RNA, like how we use DNA. The virus's strand is made up of these 4 molecules. These molecules get represented by its chemical base, either A, C, G, or U.
The way you can determine that, what virus you have, is to read the letters that make up the genetic information of that virus.
You know the way 26 letters of the alphabet come together to form words? These 4 letters combine in certain ways. Each combination is a set of instructions for the RNA cell to function. Like here, this is part of the spike protein. The spike protein shows if the virus could enter our cells or get blocked. It's not a perfect fit, but if it does enter our cells, the virus can make copies of itself, spread.
This is also the target of all of our antibodies.
Reading the genomic sequence, those combined letters, isn't easy. This is part of the original genetic code for SARS-CoV-2. The code is nearly 30,000 letters long. And here's the thing, this code constantly changes. Every time the virus spreads, some letters change or drop off. It's usually no big deal, part of the process. But when the changes happen on that spike protein, the virus may be able to enter our cells easier, even evade our immune system and vaccines. The virus itself may be weaker or stronger, but can spread faster.
There are other changes in the virus's genome outside of just spike. So there's more mutations out there. Their functional significance is not clear. Many of them could be not important.
The different mutations flow like a family tree. SARS-CoV-2's original strain mutated. The letters changed. And as it spread, it kept changing. Some family lineages got stronger, most died out. Eventually, it led to variants like the Alpha variant and the Delta variant. Genomic sequencing, reading those letters, is how we find threats. But that means we have to look.
The U.S. is currently around 4.5% to 5% sequencing of all of its cases.
Texas records show the CDC sequenced more than 6% of the state's positive cases in the last month. State, private labs, and universities like UT also run sequences, hospitals too. Baylor Scott and White, for example, looks through more than 80% of its positive cases.
The main benefit here is surveillance. So for surveillance, do you really need to check every single patient? Many experts believe 5 to 10% sequencing is quite sufficient. It's financially and logistically very difficult to sequence every single patient.
Omicron is one of 27 variants named in the world. Chances are it won't be the last. Because to stop the mutations, stop the variants, you have to stop the virus everywhere. In Austin with the KVUE Defenders, I'm Erica Proffer.